The talk today is on discussing the status of the SARS-CoV-2 specific pro and antiviral genes on the ocular surface, and discuss probably the insights into susceptibility risk and possible preventive measures. So the COVID-19 pandemic caused by the SARS-CoV-2 virus has really changed our lives and the way we look at our patients. And we do know that the cellular axis, and it was so beautifully described by Dr. Gupta about how the SARS-CoV-2 depends on the binding of the viral spike protein S to the surface receptor on our any epithelium, be it the cornea conjunctiva of the ocular surface or the retina. And the priming of this is done by the TMPRSS2 and the cathepsin L. And what we've seen is that the cornea and the conjunctival epithelium has obviously large areas of mucosa, which make it a very good area for the viral entry. The ACE2, the TMPRSS2, and the cathepsin L are predominant in the cornea, the conjunctiva, and of course the airway, because that's the primary area where the virus enters, and thus making them very good potential routes for viral entry and transmission. We do know that modulation of the ACE2 expression and its downstream effects have been reported in lung disease. But the ocular surface-related SARS-CoV-2 mediated immunopathology is poorly understood, and that's what we're going to discuss today. So this is a sort of a life cycle of the virus and how it enters into our cell, the binding, the entry, the replication. And what we need to highlight here is that basically the key players in this fight, when we are talking about trying to see how to stop this virus, is looking at how it's entering and the priming of the virus, and the three, very important ACE2, TMPRSS2, and the cathepsin L. And how is the host response, or how do we fight this virus, which is mainly by the interferons, and interferon 1 induces the MIX-A or MIX-1, which is, as we know, an inhibitor of various viruses. So the aim of the study was to study the expression patterns of genes associated with SARS-CoV-2 attachment, entry, and the host antiviral response. And this was done across groups of healthy controls, eyes from COVID-19 disease, and COVID-19 recovered patients. And the second objective was to determine whether there's any molecular changes by topical agents which are available to us, like trehalose or chloroquine, and whether this could be used for ocular surface prophylaxis. So this was a cross-sectional study which we did after the ethics committee clearance, and the full globes collected during the eye collection, which were deemed not suitable for transplantation, were used in this. And of course, very strict precautions because of the COVID situation were used for all the eye collections. So once we had received these eyes, we dissected it layer by layer, right from anterior to posterior. So corneal epithelium, stroma, endothelium, conjunctival epithelium, and so on and so forth. And then the samples were stored at -80 degrees. Some of them were used for gene expression analysis for the mRNA levels of the various proviral host genes and antiviral response genes, which we talked about. And sectioning of the histopathology specimens was done, and immunofluorescence was used for specific proteins. So these are the results. So we break up our results into 3 or 4 parts. And the first is to see how the entry and priming genes of the SARS-CoV-2 are in the ocular tissues. So on the left side, the graph basically shows the ACE2 expression, the mRNA expression across the various layers or structures in the eye, right from the RPE and the ocular surface, including corneal conjunctiva. And it's compared to primary lung epithelium that we obtained. And the red dotted lines are just to bring your attention to the ocular surface structures like the corneal and conjunctiva. And what you can see is that the quantitative PCR exhibits higher expression of the ACE2 mRNA in the cornea and conjunctiva, okay, as compared to, is compared to the primary lung epithelium. On the right side, we're looking at the immunofluorescence of the various, the structures which we sectioned. And the green color is what is marking the ACE2. And it's very evident that there's intense localization of the ACE2 in the corneal epithelium and the conjunctival epithelium, as you can see. So these are in normal ocular tissues. When we look at our next marker, which is the TMPRSS2, same we see a very high expression of the TMPRSS2 in the cornea and conjunctiva, as shown in this graph, the mRNA expression. And on the right side, again, it's the immunofluorescence of green color. And you can see that there's intense localization in the corneal epithelium and conjunctival epithelium. So no doubt there are a lot of localization of these very important structures to which the virus binds on the ocular surface mucosa, making it increased and very susceptible to the binding of the virus. Now coming to our host response. So the antiviral response by the interferons, we found, again, we looked across all the various structures through the eye, as discussed in the previous slide, and we found that there was definitely a high expression of the interferon receptors, but the gene which it induces, the MICA, which is actually a protective factor for our ocular surface, was much lower in the ocular surface tissues compared to the other ocular tissues. So it implies that there's probably a lowered protective response that we have. Now, when we go forward and look at a comparison of the normals versus a person who is actively having COVID versus a COVID recovered eye. And these are the three columns of images that you see in the immunofluorescence. Then here the ACE2 is depicted by a red color and the MICA is in the green color. So I draw your attention to the fact that the ACE2 has pretty similar levels in the corneal epithelium, whether it's a control cornea or an active COVID-19 positive eye versus a recovered eye. But the MICA, which is our protective factor, again, is much lower in the eye which was from a person diseased with an active COVID-19 disease compared to both the controls or the COVID-19 recovered eye. And just to bring it, highlight that a little more, we see that the mean immunofluorescence intensity, of course, was much lower in the COVID-19 positive eye of the MICA. So when we look at the access routes, so till now we were, again, talking about those same factors which are so important for us. Again, we find that here the TMPRSS2, which is in red, and the cathepsin L, which is in green. So the expression of both of these were much higher in the COVID-19 positive eye as compared to control and the COVID-19 recovered eye. So just if you can look at this image itself, it's so representative of what we are seeing. And this is just to reiterate the fact that the ocular surface seems to be so susceptible to the viral entry. And definitely once there's an infection by the COVID-19, there's a definite change as compared to a normal and or a recovered corneal tissue. So what happens? So this is in an ocular surface which is presumed normal. But when we have an altered ocular surface, for example, in dry eye disease, we wanted to know if there's any change from this normal protective mechanism or normal receptors that are available. And so we did this experiment of impression cytology, which we had collected pre-COVID time of patients of dry eye disease. And very interestingly, we found that in a patient with an altered ocular surface, like in dry eye, there's a definite increased expression of those three, the ACE2, the TMPRSS2, and the cathepsin L. And there is reduction in our protective interferons, the type 1 interferon. So definitely this altered balance could imply that it increases the susceptibility even more. So with this background, it didn't end there. We took it further to see if there's anything more we can do. And I invite Dr. Swaminathan to talk about this work further.
Thank you, Dr. Sharon. And one of our primary objectives is to find out whether we have any good prophylaxis for the ocular surface. And we chose chloroquine and trehalose based on their literature on how they would be beneficial in bringing about antiviral response. In the next slide, what you would see here is going to be an in vitro assay. We have taken human corneal epithelial cells and human alveolar epithelial cells and treated them with trehalose and chloroquine for a period of 24 hours. What you can see in the table is a representation of what you see in the graph towards the left is a significant reduction in ACE2, TMPRSS2, and cathepsin L can be seen by both trehalose and chloroquine. However, what was startling was trehalose was able to induce type 1 interferon, whereas chloroquine was not only able to induce, but it actually showed a reduction in the type 1 interferon expression. So we thought, in order to go further to test the efficacy, we chose trehalose, and we chose to do it in humans. We had very enthusiastic volunteers who wanted to contribute towards the knowledge and bringing about COVID prevention. So we had a group of individuals who took trehalose eye drops for a period of 10 days, 2 times a day. And we had impression cytology and nasal swab from the same individual before the start of this treatment and day 10 as well at the end of it. What you see there in the graph has been divided into ocular mucosa via impression cytology and nasal mucosa expression using the nasal swab. What you can observe and appreciate is a significant reduction in ACE2, TMPRSS2, cathepsin-L in the ocular surface mucosa, and an induction in interferon alpha, and that is type 1 interferons. And there was a bit of reduction in the ocular mucosa of interferon gamma, that is type 2, and induction of type 3 interferons, that is interferon lambda 1. So what you can see in the summary is that trehalose in an individual was able to induce the beneficial alterations in the ocular surface mucosa as well as the nasal mucosa. But you might wonder what was happening in the non-treated group. In the next slide, you can appreciate that in the untreated control, between day 0 and day 10, we don't see any such changes whatsoever. So what do you have in hand is a potential prophylactic, which Dr. Sharon can elaborate on how it can be used clinically.
So what we knew is, of course, that the ocular surface and the conjunctiva is a potential route for viral entry. And we also knew of the existence of the ACE2 and its how it increases the risk of infection. But what we're trying to add is the expression patterns of other various attachment or entry related genes in normal human ocular tissues, and also compared it to those tissues obtained from COVID-19 infected and recovered subjects. We also showed that unfavorable ocular surface, like for example, in dry eye patients, could also change how all these factors behave. The potential use of trehalose as a prophylactic eye drop is important, as it could reduce the expression of the attachment and also enhance the type 1 interferons, so thereby preventing the entry or transmission of these viruses. Of course, repurposing medications may help us fight in a very emergent problem. So of course, the limitations could be a smaller sample size. And of course, this is right now experimental, and we have a small pilot human study. But we hope to take this further. And these are our references. And of course, a huge round of applause and thanks to the entire team at GrowLab, led by Dr. Ghosh and Dr. Swaminathan, and thanks to Mr. Vishnu and Dr. Trilokanath, who have worked very hard with these experiments.